Okay, so welcome everybody to this In-Depth Academy session. So we have two speakers in this session today, and I'm going to be hosting the session. So you probably know how it works by now. During the talk, you can put your questions in the chat box, and then I will read them out for you at the end of the talk. So I think we can start straight away. So our first speaker of this session is Sara Farrona. Sara is a group leader in the National University of Ireland in Galway. And her main interest is Polycomb regulation. And she's going to be telling us a little about it, and also in relation to nuclear organization. So I'll pass it over to you, Sara. I think you, yes, all is good. Looking forward to
Okay.
your
Thank you very much, Stephanie, and thank you, the organizers, for giving me the opportunity to show you this project that we just started about PWO proteins and the evolution of these proteins. But just before starting with the project itself, I wanted to show you how the project started. And that occurred in an in-depth meeting, the one in Prague in 2019. And I was in a lunch break with Daniel Suber and Eva Moskova, and we were just chatting there, and we realized that we had a common interest about in the evolution of our favorite group of proteins. And from that conversation in depth in that meeting, so we have now developed this new project that we call EvoPWO. And Ahmed Khan is a postdoctoral researcher in Eva's lab that I'm also co-supervising, that is now developing this project. But everything started there in that meeting. And Ahmed got a Marie Curie individual fellowship just a few months ago, so we are very happy that he will keep developing this project in Eva's lab. Okay, now I'm ready to talk about the project itself, and this is about PWO proteins. So here you can see the 3 PWO proteins in Arabidopsis thaliana, and we call them like that because of this long name that is PWWP, domain interest of polycombs. And the first part of the name comes from the conserved domain in these proteins, that is this domain that you can see here. That is it's a domain that we saw that in PWO1 is able to interact with histone 3. And the last part of the name, keep it in mind because I will come back to that later in my talk. So the single mutants affected in each of the genes that encode these 3 proteins, as you can see here, they have very mild, weak phenotypes, very similar to wild type. They are happy plants. But if we combine these 3 mutations in a triple mutant, you can see that the phenotype is now very severe, with plants that are highly affected and just dying a few days after germination. And one single of the PWO genes, here you have PWO1, is enough to complement the strong triple mutant phenotype, indicating that probably these 3 proteins have highly redundant functions in the plant. So if we put the 3 proteins and we compare them with the other proteins in Arabidopsis thaliana that contain this conserved domain, you can see that in the phylogenetic tree they are coming together in a specific clade. And if we compare and do an alignment of the domain, of the conserved domain, you can see that is very well conserved. And also we can use these in silico tools to predict the structure of the conserved domain. And as you can see here, just the one for PWO1 against a protein that has been crystallized, you can see that the domain is probably folding in the expecting 3-dimensional structure for this type of domains. And what is nice about this conserved domain is that allow us to look for other PWO proteins through evolution, basically. And the first thing that we realize is that we only identify PWO proteins in plant species. And these are many different plant species. I guess that is very small and you cannot see the names, but believe me, these are many different plant species in which we can identify PWO proteins. And as you can see here, the sequence of the domain is very well conserved, that helping us to identify these proteins. But we don't find them in any other organisms. They are plant specific, so they are not, for example, in animal or in yeast. So using this type of alignments, we can build a phylogenetic tree. And as you can see here, there are 3 main clades in this tree. One of the clades is for the PWO1-like proteins. The second clade is for the PWO2-3-like proteins. And then we have a final and more ancestral PWO clade in which we have the most ancestral PWO proteins, and these are the ones coming from fern and fern-allied species of Lycophytes. And in this is the group in which we identify the most ancestral PWO proteins, because these are Selaginella species are the ones that we identify, the most ancestral species in which we are able to find the PWO protein. So our hypothesis is that the PWO proteins may have origin in this time during the evolution of plants. And this is a very interesting moment in the evolution of plants, because it's when plants are moving from aquatic environments to terrestrial environments, and they needed to acquire many different traits to allow them to adapt to this environment. So we are trying to see if the PWO proteins have contributed to the evolution of plants and to help plants to adapt to new environments. We can also have a look not just at the protein, but also to the gene of this encoding these proteins. And as you can see here, just marked in blue, you can see the PWO1-like genes, or encoding PWO1-like proteins. And well, the first thing that you can see in all of them, in all of these genes, is that there is an intron separating the coding sequence for the domain, for the conserved domain. So as you can see, the conserved domains are then splitting between exon 1 and exon 2 by this intron, and it's happening in all the PWO genes, very highly conserved. And then when we look at this, the PWO1-like genes, we have another 2 extra introns, as you can see here, and this is also very well conserved in the other PWO1-like genes. Okay, and then for the PWO2-3-like genes, we usually have just one extra intron, so 1 and 2 introns in total, as you can see in all the genes that are marked in white. PWO2 is in Arabidopsis thaliana is just an exception to this rule, and we have an extra intron. But in the other genes that we have, the PWO2-3-like genes that we have identified, there is only two introns. So if we look at the most ancestral genes that are these from Selaginella mollendorfi, we have a protein that we call PWOA and a PWOB protein, but when we look at genetic level, we see that the gene encoding PWOA has 3 introns and the one encoding PWOB has 2 introns. So this could indicate that PWOA is more related to PWO1 and PWOB is more related to PWO2-3 proteins, but obviously this is a hypothesis that will also require further investigation. I told you at the beginning that I was coming back to the second half of the PWO, that is the interaction to Polycomb proteins, because indeed we identify PWO1, the PWO1 protein in Arabidopsis as in a Gis2-hybrid screening interacting with the Carly leaf protein. And Carly leaf is one of the histone methyl transferases of this complex that is PRC2 for Polycomb repressive complex 2. And also by Gis2-hybrid, we checked that PWO1 was indeed able to interact not just with Carly leaf, but also with Swinger and Medea, that are the other 2 histone methyl transferases of this complex. And in Planta, we also confirmed the interaction with Carly leaf using co-IP experiments with a PWO1 GFP Carly leaf and cherry transgenic line. So which complex is PRC2? So PRC2 is the complex that is able to place this histone mark that is 3 methyl groups on the lysine 27 of the histone 3. This is epigenetic mark that is involved in repressing expression of genes, and this is a euchromatic mark, so genes that have to be repressed but also dynamically regulated. And indeed is a very important epigenetic mark because around 20% of all Arabidopsis genes, and also this has been confirmed in all the plant species, are marked by this histone mark. And I told you before, there are 3 proteins able to introduce the mark in this complex, the Carly leaf, Swinger, Medea. Doesn't mean that we need the 3 together. One of them is enough, and we have indeed different PRC2 subcomplexes. PRC2 has a very important role in regulating development because among these genes, these 20% of K27 mark genes, we have key developmental genes, and PRC2 is regulating indeed the main developmental switches in the plant. And when we have a strong PRC2 mutant, we don't have a proper plant anymore. We have something that develop like forming a callus-like structure. So as I told you, PWO1 was identified interacting with these proteins. So we cross the two mutants, Carly leaf and PWO1, and we identify these phenotypes. That means that PWO1 is just enhancing the Carly leaf phenotypes that, as you can see here. So we analyzed if that was maybe because it was affecting the activity of the complex of PRC2, just looking at the position of the K27 mark. And as you can see here, checking using some of the typical Polycomb target genes, we identify a decrease in the presence of the mark. So we thought, okay, PWO1 is required for the activity of the complex, but then we look at the level of histone 3, that is the substrate to add this mark. And as you can see here, we also identify a significant decrease in all the genes that we test for the presence of this histone. So that probably means that PWO is not just directly involved in the deposition of the mark, but probably more related to the deposition of the histone 3, or at least to the occupancy of histone 3 at set of genes. But I was trying to focus my talk in evolution, as I told you at the beginning. So if we now put that the PRC2 complex that is interacting with PWO into the evolutionary tree that I showed you before, so what we see is that PWO is much well conserved than PWO proteins. So we have components of the PRC2 all over the plant kingdom. And I think they are also present, for example, in algae. And I think that is something that Eva will show you later. So I don't want to go too much into detail on that. But I just wanted to show you that they came, the PRC2 proteins came before PWO. So we are very interested in understanding what PWO proteins brought into the evolution of the Polycomb pathway and how PWO proteins may have contributed to shape the activity of this complex and probably contributing to reprogram the nucleus through the modification of the activity of the complex. So what we have done so far is just taking the PWO protein from Selaginella, that as I told you is one of the most ancestral PWO proteins that we have identified. We used these two hybrids to see if this protein was still able to interact with proteins of PRC2 from Arabidopsis thaliana. So we are now mixing ancestral PWO proteins with modern PRC2 proteins. And as you can see here in the GIS2 hybrid data, the ancestral PWO protein, the Selaginella PWO proteins, are still able to interact with Carly leaf and Swinger, okay? And probably the Swinger interaction is a bit stronger than with Carly leaf, but there is still interaction. Obviously, this is just a very preliminary first result, and we need to confirm this result in plant, and this is what we are still working on. Okay, so this is in the interaction with PRC2, but PWO proteins are able to interact with other proteins, and I'm going to show you something about that. So here you have the localization of the PWO1 protein in tobacco cells. So these are transient assays in tobacco epidermis, in tobacco cells from the epidermis of tobacco leaves. And something that we realized is that we, the first important thing is that PWO1 is a nuclear protein and is localizing over the nucleus, but it's also forming speckles, as you can see here. We see 3 speckles. And something that we realized, it was just an observation in the lab, is that these speckles have a tendency to be more in the periphery of the nucleus. So we decided to quantify that, and indeed, we quantify this observation, and you can see here in this graph that there is more speckles, PWO1 speckles, always in the nuclear periphery. We also did CoIP analysis to try to identify the proteins that were interacting with PWO1 using CoIP experiments coupled to mass spectrometry, and we realized that more than half of the proteins that were coming in our PWO1 CoIP have been already related with the lamina or with the nuclear envelope. So we decided that we have these two data put in PWO1 somehow in the nuclear periphery, probably interacting with the nuclear envelope of the lamina. So let's go a bit deeper into that. So we're focusing one of those proteins that is crowded nuclei 1 or CRAN1 that we have seen already several times in this meeting. And we, for example, this CRAN1 mutant, we saw it yesterday, and it was very nicely described. But here I just want to show you that it has this phenotype with a smaller nuclei. So because of that, and because of this interaction, we decided to look also to PWO1 nuclei, and we realized that the nucleus are also smaller. And this is something that probably we haven't realized, or it would have taken us longer time to realize if it's not because of this interaction. And so we decided also to look at the transcriptomic data and to combine the data from PWO1 mutant with CRAN1-2. We focus in the double mutant just to enhance a bit the phenotypes. And we cross this data. We focus in the regulated genes in our transcriptomic data, and cross the data with the K27 target genes, and only focus on the upregulated, because if you have changes and a decrease in this mark, you expect that the genes are upregulated, basically. As you can see here, there is a good number of genes between the genes that are upregulating PWO1 and PRC2 target genes. The same for CRAN1-2 upregulated genes. But when we look at the three together, there is a small set of genes only, 36 genes. But when we look at these genes a bit closer, you can see that most of the genes are involved in response to stress, what could indicate that these genes are genes that have to be maybe dynamically regulated and able to respond quickly, for example, to any stress. Let's put evolution again, and if we look at the evolution of proteins in the lamina, particularly to CRAN proteins, and here you have this nice phylogenetic tree from this paper, a paper that is coming from Christophe Tatou, David Evans, and Aline Prob's labs, all of them in InDepth also. And you can see that the most ancestral proteins are coming here in Fiscomitrella patterns. And we also just confined this data, and we found exactly the same. So here are the most ancestral CRAN proteins, again from this case from mouse, and we have two, A and B. And it seems that the most ancestral proteins are more related with the CRAN4 clade, because the phylogenetic tree is divided in two big clades, the CRAN1 clade, that is CRAN1, 2, and 3 type of proteins, and then CRAN4 proteins in a different clade. So the most ancestral could be that. And remember that I told you that PWO1 has been related, was found interacting with CRAN1 in our co-IP data, but we also found there CRAN4, Anyway, this is something that Kalyani is going to show you much better and with a much level of detail in a talk this afternoon. So I would like to encourage you also to join her talk this afternoon. But again, coming back to evolution, if I put now the three components together in our evolutionary tree for the plant kingdom, you can see here PWOs coming probably at this time in the evolution of plants, then PRC2, as I told you, much earlier, and then CRAN proteins just coming just a bit earlier than the PWO proteins in moss. So we are highly interested in this project in understanding how these three components has been interacting through evolution and affecting each other throughout the evolution of plants. And this is what is finally bringing me to the EvoPWO synthetic biology project. That is this project that we just started a few months ago, in which we are playing with the evolution of plants and with the ancestral and the modern version of the proteins that I introduced for you. And we are just going up and down of the phylogenetic tree. So our idea is to take the most ancestral proteins to complement the corresponding mutants in Arabidopsis thaliana, using Arabidopsis as our modern plant species. And then also taking the Selaginella PWO ancestral species to not only move them into the modern species, but also into a more ancestral species that is PWO-less, that it doesn't have a PWO protein, and let's see what this is bringing into moss, and if we are able to reprogram the nucleus, because our current model is that PWO1, at least within the PWO, the PWO1-like proteins could be acting as a link between the nuclear periphery and the Polycomb pathway, at least for regulating a set of genes. And this is what we want to see, how this appears through evolution. Okay, so this is just the end. I would like to say thanks to everyone in our 3 labs, my lab and also Eva's and Daniel's labs, because of the contribution, and particularly to Ahmed Khan, that is the person that is really developing this project in Eva's lab. And also just remind you about Kalyani's talk this afternoon, and because he's going to talk more about PWO proteins, about the role of PWO proteins in the nuclear peripheries. He's going to explain that better than me for sure. And obviously acknowledge InDepth, because we met in that meeting, and that was really the starting of this project. Otherwise, I think that the project will stay waiting in a drawer in my office. So that's really great that it gave us this opportunity. And thank you very much, everyone. Happy to take questions.
Thank you very much, Sara, for this very interesting and nice to see also how these new collaborations have started with the InDepth. So if you have any questions, I'm sure there are questions, so don't be shy, write them on the chat box. Otherwise, I have some questions. In fact, I have several questions. I could start with that. So, I mean, I was just, thinking of already from the beginning, you showed the differences in the intron of the different PWO1 like genes. Do you have any idea? What is the idea what this difference could be associated with? Any particular difference?
No, but not really. But we look at that because the ancestral proteins are coming in just one clade in the phylogenetic tree, and we have 2 already in the most ancestral species, in Selaginella species. So we couldn't know which one corresponded to what, because it was already coming in a different clade in the phylogenetic tree. So there was that was just the reason to look at the genetic level to see if there was something there that could indicate if one of the protein was more PWO1 or more PWO2/3. And we look at that because of this reason. But why that structure is so well conserved, why we have always that intron split in the conserved domain, for example, and why we have already 2 proteins from the most ancestral species. And yeah, it's something that we don't know yet. Yeah, I'm just asking because we've been now more and more interested in the regulation through the intron. So I was really, I know, yes.
Maybe you're having some idea this could have a function.
Yeah, well, we only know that in Arabidopsis thaliana, that we have 3 PWOs, PWO1, 2, and 3. We always, cloning PWO1 and 2 was no problem, but PWO3 was a nightmare. And we always thought that it was because of different type of transcripts and that are not well annotated, maybe. And that could maybe indicate a role for, at least in PWO3 and Arabidopsis for the introns, but that's the only data that we have so far.
Okay, so now the first question is popping in by Konstantin. Do you find chaperones associated with PWO since you detect different H3 occupancy?
And not chaperones, we identify chromatin remodeling proteins. And this is something that Kalyani is going to show in the afternoon. So we identify, for example, SWI/SNF proteins, and also the proteins that have been involved in remodeling chromatin. And we also identify histones there. Okay, so again, confirming the role of PWO1 and the ability to interact with histones.
So related to this H3, so you see a difference in occupancy or in the H3 amount, H3?
What we see there is just amount in our chip PCR data.
Is it overall or specifically in these genes? Did you do just
in these genes? Yes, only in the genes. And this is something that we are still are trying to investigate deeper. Yeah. So we don't know how general is this role, and is this just because the decrease is because of the changing in the position of the nucleosome, or just because of a decrease in the amount of nucleosome? This is something that we don't know yet.
Okay. And I mean, it's quite interesting, right, to see this, that there's this new protein there that is associated just with plants. So now you seem to be picking up something related to stress. So is it what the idea is that in this transition to the terrestrial environment, you would need an additional regulation because now you have perhaps a more variable, more stressful environment?
Yeah.
Is that the idea, or?
Yeah, this is a very beautiful hypothesis that we are considering, because obviously, I mean, we know that PWO1 is together with PRC2 and crowns are involved in controlling development. So what we like to see if they help plants to bring new developmental traits that help them to adapt to these new environments. So that's the idea of not only putting the proteins from ancestral to modern species, but also from ancestral to more ancestral species that don't carry the proteins, to see if we now, I don't know, mess up with the development and we have new traits, and we can get a hint of what was the role of these proteins throughout evolution. And yeah, that could be a nice hypothesis. And also, as you said, because stress, obviously adapting to moving from aquatic to terrestrial environment was probably a huge stress for the plant, and we know, and we have those genes that are regulated by the three components that are responding to stress, so that could indicate also a role of these proteins through evolution in regulating these genes. But these are the hypotheses, and it's part of the EvoPWO project. Yeah, basically.
No, it's really interesting, I think. Now I have just one more question. I think we still have time. It's more technical. It's related to the localization of these PWO foci. They are really beautiful, but they are very different from Polycomb localization, which is normally you would see throughout the nucleus, right? So they don't form this kind of huge blobs in the periphery. So why? So you think there is the colocalization with Polycomb, but just not everywhere? Is that what you're thinking? I'm thinking it's so different.
Well, these are again tobacco cells. Okay, so I think that we discussed quite a lot yesterday about the tobacco transient experiments, and but when we put, for example, a protein from the PRC2 like curly leaf together with PWO1 in tobacco, we see both colocalization in diffuse colocalization in the nucleoplast plus the speckles. But PWO changed the localization of curly leaf. Curly leaf is usually just in the nucleoplast, and then you put PWO1 and forms also the speckle, perfectly colocalization with PWO1. So our idea is that PWO1 is the one bringing also curly leaf to those speckles. We know that also PWO1 can dimerize or can interact with itself, so probably can dimerize, so that could maybe help to form these aggregates. However, when we look at Arabidopsis thaliana, the localization of PWO1 is slightly different. So is again is forming aggregates, but not this kind of nice speckles. Okay.
But so is with curly leaf then, or is harder to assess?
We didn't check the colocalization in Arabidopsis with between the two. Yeah. We check. I think that we tried colocalization with K27, but and there was some colocalization with the mark, but these experiments were tough, basically, and. Yeah, so it's much more difficult when you go to Arabidopsis thaliana and and analyze the localization of the protein. Yeah.
So Konstantin just put now a comment, maybe PWO target polycombs to new sets of genes. Maybe he can unmute himself and explain what he means.
Yeah, this is just like maybe a little bit of summarizing of what was said, because it kind of like Polycomb, as far as I know, it kind of has these several complexes which have these particular functions with Polycomb, and yeah, just maybe it's kind of was necessary to evolve this protein to target some new set in these like terrestrial plants. So.
Yeah, exactly. So that's our idea, that because PWO1, we see PWO1 as kind of a reader of chromatin, because it has this conserved domain that is able to interact with histones. And the rest of the protein, I forgot to tell you that, but it doesn't have any conserved domain, and it's predicted to be highly disordered. Okay. And that means that it's maybe kind of promiscuous to interact with many other different proteins, thanks to this disorder, organization, you know. So it could be like kind of a reader and then recruiting proteins to the right place. But obviously, this is also part of the hypothesis. Yeah. So thanks a lot. Very nice discussion. Thank you very much.
Thank you very much for your nice talk.